Hello everyone, my name is Natalia Islam and my topic for this presentation is a study of the 2023 Western Equine Encephalitis outbreak in South America. We will now discuss some key information about Western Equine Encephalitis. The Western Equine Encephalitis virus is an alpha virus belonging to the Togaviridae family. It is commonly found in rural agricultural areas and small towns. The transmission of Western equine encephalitis virus is divided into two interlinked transmission cycles, the enzootic or endemic transmission cycle and the epizootic or epidemic transmission cycle. The enzootic cycle occurs during spring and early summer. The mosquito vector Culex tarsalis feeds almost entirely on birds which act as a reservoir for the disease and make sure it is constantly present in the host population. During the summer, the Culex tarsalis expands its host range and feeds on humans, horses, and rabbits. The Aedes melanimon species of mosquitoes feed on these infected rabbits and equine. A secondary cycle or epizootic cycle involving Aedes melanimon and small mammals develops, which may transmit the infection to humans and equines such as mules, horses, and pheasants. The Western equine encephalitis virus cannot be transmitted through aerosols. However, it can be transmitted from mother to fetus. If viral load is high enough, it can also transmit through blood transfusion. Infection with Western equine encephalitis virus usually presents as a subclinical infection, with patients exhibiting symptoms such as fever and chills, nausea and vomiting, headache, and neck stiffness. While some patients may recover spontaneously, others can develop encephalitis. This neuroinvasive disease develops acute inflammation of the meninges and brain parenchyma and presents in patients as somnolence, confusion, coma, and death. Adults have a better prognosis compared to infants, young children, and older adults, as the latter are more likely to develop neurologic complications. In fact, older adults are most likely to die from complications, and young children are most likely to develop permanent neurologic disability. This disease also presents as central nervous infections in equines, causing blindness, staggering, and seizures, with high case fatality rates often leading to death within days. Cases of Western equine encephalitis have been reported since the 1930s. The largest outbreak to occur was reported in the 1930s to the 1940s. This outbreak resulted in greater than 3,000 human cases and tens of thousands of equine cases. Western equine encephalitis cases in the United States began to decrease from the 1960s, with less than 700 reported cases. There have been no reported human cases since 1999 in the United States since. South America saw similar outbreaks in the 1970s and 1980s as North America, followed by isolated cases in Argentina in 1996 and Uruguay in 2009. Canada has also seen outbreaks, a large one in 1941 and later outbreaks in the 1980s. This map on the right shows the number of reported human cases of Western equine encephalitis by state since 1964 in the United States. The highest number of cases were observed in California, Colorado, North Dakota, and Texas. In December of 2023, that is just last year, a large outbreak of Western equine encephalitis was seen in Argentina and Uruguay. This outbreak only ended this year in April 2024. The outbreak began in northeastern Argentina, spreading to central regions and Uruguay. The outbreak resulted in 112 human and 127 equine confirmed cases, and 105 human and 2,421 equine suspected cases, with equine cases preceding human cases in both countries. This presentation aims to discuss this article, Molecular Epidemiology of Western Equine Encephalitis Virus, South America 2023 to 2024, where the Western Equine encephalitis outbreak in Argentina and Uruguay is discussed, and the circulation of Western equine encephalitis virus is investigated in neighboring countries such as Brazil. Moving on to the methods used in this study, there were quite a few molecular techniques that were used in each step. First, Epidemiologic data containing demographic and clinical characteristic of Western equine encephalitis cases in humans and equines in Argentina and Uruguay were obtained from the Pan American Health Organization. Next, infected equine and mosquito samples were collected. Brain tissue samples were obtained from 31 fatal horse cases which had been submitted to the Center for Health Surveillance of Brazil. Mosquito samples were obtained from mosquito pools collected from areas with a recent history of neurologic equine disease. Both samples were screened using a molecular method called RT-PCR. RT-PCR is a type of PCR. PCR stands for polymerase chain reaction. It is a molecular method which uses a sample of tissue, blood, or other body fluids to test for the presence of certain genes or molecules that may be an indication of a disease. In this study, the samples were used to screen for the Western equine encephalitis virus. In addition, it was also used to screen for Eastern equine encephalitis, West Nile, St. Louis encephalitis, Mayaro, and Oropoche viruses. Equine brain samples were also screened for the rabies virus. The 3 out of 31 equine brain samples that tested positive for the Western equine encephalitis virus were sequenced using a hybrid capture-based metagenomic approach, resulting in the establishment of 3 novel Western equine encephalitis viral horse genomes. This was done through molecular sequencing, which is a technique that can be used to determine the entire genetic makeup of an organism. This technique is used to find how diseases develop, how they can be diagnosed, and changes in areas of the genome. Lastly, the 3 novel Western equine encephalitis viral genomes that were developed were aligned with Western equine encephalitis viral strains with complete coding sequences that were available in the GenBank database. This was used to generate a phylogenetic tree. Phylogenetic analysis uses nucleotide or amino acid sequences or three-dimensional structure domain sequences to construct an evolutionary tree that shows the evolutionary relationship among different taxa at the molecular level. Phylogenetic analysis can also be used to investigate domain relationships within an individual taxon. In this study, phylogenetic analysis was used to generate a maximum likelihood phylogenetic tree. Now let's discuss the results from the study. This is a figure showing the Western equine encephalitis cases in Argentina, Uruguay, and Brazil. Part A demonstrates the cumulative Western equine encephalitis cases in Argentina and Uruguay reported to the Pan American Health Organization. Part B demonstrates the locations of deaths of the 3 horses that had tested positive for the virus during RT-PCR. All 3 of these horse cases showed signs of neurologic disease and none were vaccinated against the virus. In addition, these 3 horses were also positive for rabies, but negative for all the other viruses that they were tested for. The cause of death of the other fatal horse cases were unconclusive. This is the maximum likelihood phylogenetic tree of the 3 novel Western equine encephalitis strains that were obtained from Brazil. This box shows the 3 novel Western equine encephalitis strains circulating in Brazil that were sequenced from the 3 horses positive for the virus, which are these 3 highlighted strains in the box, alongside 6 other strains obtained from Uruguay. All 9 of these strains formed one independent cluster. This cluster is related to the 1958 CBA87 strain from Argentina, as it shares 94.9 to 97.6% nucleotide identity with the 1958 CBA87 strain. This makes sense as they have the same ancestor. It is hypothesized that the new lineage with the 3 novel strains had evolved and become independent many decades before from the strains circulating in North America. Results also show that the virus has been circulating South America since 2009. There may be several reasons why Western equine encephalitis cases have not shown up from 2009 to 2023 in South America, such as limited enzootic circulation of the virus between vectors and reservoirs, or lack of disease surveillance, especially in rural areas where cases are more prevalent and more likely to go unnoticed. The last reported case in South America appears to have been April 2024. This may be due to the increased immunization of horses. In conclusion, the study identified an active Western equine encephalitis virus circulation in Brazil and a novel viral lineage associated with fatal cases in horses. These findings stress upon the fact that there is dire need for continuous laboratory diagnosis and surveillance for the virus in both humans and horses. More ecologic studies are also needed to help us understand the transmission dynamics and the ecologic drivers that led to the reemergence of the disease in South America. Lastly, the authors stressed on horse immunization being considered a way to alleviate the effect on animal health. With the reemergence of the Western Equine Encephalitis virus and discovery of a novel lineage, it is important that we plan our next steps carefully. We need to increase surveillance of viral strains, especially in rural areas. New diagnostic and therapeutic interventions are needed to prevent complications associated with the disease. Currently, there is no FDA-approved preventative or therapeutic medical countermeasure against the virus, except the TSI-GSD210 vaccine that has been in clinical trials since the 1970s. In the meantime, educating high-risk populations about preventing mosquito bites and handling horses safely is an effective way of mitigating spread of the disease. In addition, professionals that work with equine must be sought out. Since immunization against the virus is based on a risk-based assessment of the horse's potential exposure, it is important to have practitioners to consult about what is best for horse and human health. Thank you for listening to my presentation.